Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we're going to go over some practice questions regarding the plasma membrane and cell-cell junctions. Feel free to pause in between, and with that, let's get started. Number 1. A student is trying to determine the type of membrane transport occurring in a cell. She finds that the molecule to be transported is very large and polar, and when transported across the membrane, no energy is required. Which of the following is the most likely mechanism of transport? A. Active transport, B. Simple diffusion, C. Facilitated diffusion, or D. Exocytosis? Okay, so let's work through it. A. Active transport. Well, there's no energy required, so it can't be active transport. B. Simple diffusion. Well, since the molecule is very large and polar, it can't be simple diffusion because those have to be very small molecules or uncharged molecules. And C. Facilitated diffusion. Facilitated diffusion is a type of passive transport, so it requires no energy, and it transports larger molecules. So the correct answer would be C. Let's just talk about why it isn't D though. D is exocytosis, and as you may recall, exocytosis requires energy. Number 2. A researcher treats a solution containing animal cells with ouabain. This is a poisonous substance that interferes with the sodium-potassium ATPase embedded in the cell membrane, and as a result, the cell lyses. Which of the following statements best describes ouabain's effects? A. Treatment with ouabain results in high levels of extracellular calcium. B. Treatment with ouabain results in high levels of extracellular potassium and sodium. C. Treatment with ouabain increases intracellular concentrations of sodium. Or D. Treatment with ouabain decreases intracellular concentrations of sodium. So let's think about this question. To answer this question, we don't really need all this background. All we need to know is the cell lyses. Now, if we know the cell lyses, what do we know? We know that water is coming in, which means that solute concentration has increased in the cell. Now, this could be either meaning that the intracellular concentration of a solute is increasing, or the extracellular concentration of a solute is decreasing. There is no options choice that says that the extracellular concentration of a solute is decreasing, but there is one that says the intracellular concentration of sodium increases, and that is C, and that is the correct answer. Number 3. Which of the following is not a function of the cell membrane? A. Cytoskeletal attachment, B. Protein synthesis, C. Transport regulation, or D. Second messenger reservoir? This should be an easy one. It's protein synthesis, answer B, because they just get put onto the cell membrane, they don't get synthesized there. Number 4. The dynamic properties of molecules in the cell membrane are most rapid in A. Phospholipids moving within the plane of the membrane, B. Phospholipids moving between the layers of the membrane, C. Proteins moving within the plane of the membrane, or D. Proteins exiting the cell through exocytosis. And this one is A. Phospholipids moving within the plane of the membrane. They don't move between the layers too often. It's not as often as them moving side to side, and proteins don't move as often as that either. Most are embedded, so they don't even move, and they don't exit the cell often either. So, A. Number 5. Which lipid type is the least likely to contribute to membrane fluidity? A. Unsaturated glycerophospholipids, B. Transglycerophospholipids, C. Cholesterol, or D. Unsaturated sphingolipids? So let's talk about trans fats, right? Trans fats, they have a double bond, or maybe more, but since it's in the trans position, that doesn't create the biggest of kinks. And this actually increases the melting point, and by doing that, it decreases membrane fluidity. The other ones, which are unsaturated fatty acids, unsaturated glycerophospholipids, cholesterol, and unsaturated sphingolipids, they all increase membrane fluidity by a lot. So this one isn't exactly the most correct answer, but it is the most right answer. And so the option that is correct is option B, transglycerophospholipids. Let's move on to question 6. A membrane receptor is most likely to be A. An embedded protein with catalytic activity, B. A transmembrane protein with sequestration activity, C. Membrane-associated protein with sequestration activity, or D. Transmembrane protein with catalytic activity. Now, we know that it must be a transmembrane because it's a receptor, so it must get to the other side, and they activate a secondary messenger. And so to do this, you have to have catalytic activity to act as an enzyme to activate something else. So it must be D. Sequestration just means you hold on to it, and this is not what they're doing. They're not seizing anything. They're just passing on the message, so it's D. Moving on to 7. Which of the following is not a cell-cell junction in animals? Desmosomes, gap junctions, plasmodesmata, or tight junctions? This should be easy. It's plasmodesmata. These are not in animals. These are in plants. Number 8. Which of the following is true of diffusion and osmosis? A. Diffusion and osmosis rely on the electrochemical gradient of only the compound of interest. B. Diffusion and osmosis rely on the electrochemical gradient of all compounds in a cell. C. Diffusion and osmosis will proceed in the same direction if there is only one solute. Or D. Diffusion and osmosis cannot occur simultaneously. So let's work backwards here, starting with D. Diffusion and osmosis cannot occur simultaneously. Well, this is wrong because they can occur simultaneously. There's nothing stopping them. C. Diffusion and osmosis will proceed in the same direction if there is only one solute. No, they will proceed based on the concentration gradient. B. Diffusion and osmosis rely on the electrochemical gradient of all compounds in a cell. No, it's not this one, because as we know, each compound has its own concentration gradient. And if they were all going based off some other compound's concentration gradient or in a general concentration gradient, that would not be good for the cell. So the option choice that is correct is A. Diffusion and osmosis rely on the electrochemical gradient of only the compound of interest. Moving on to number 9. The bulk movement of liquid into a cell through vesicular infoldings is known as phagocytosis, pinocytosis, exocytosis, or drinking. This one is simple. It's movement of liquid through the vesicular infoldings, so it has to be pinocytosis. Number 10. Which of the following statements conflicts with the fluid mosaic model? A. The cell membrane is static in structure. B. Membrane components can be derived from multiple biomolecules. C. Hydrophobic interactions stabilize the lipid bilayer. Or D. Proteins are asymmetrically distributed within the cell membrane. And it should be easy. It's a fluid mosaic model. The cell membrane is not static in structure. The right answer is A. Okay, number 11. Which of the following statements best describes the physical characteristics of phospholipids? A. They're negatively charged at pH 7 and therefore entirely hydrophilic. B. Hydrophobic. C. Partially hydrophobic. Or D. Positively charged at pH 7 and therefore entirely hydrophilic. This is another easy one. It's C. Partially hydrophobic. Okay, number 12. If the percentage of unsaturated fatty acids in a membrane is increased, will membrane fluidity increase or decrease at body temperature? So as we know, unsaturated fatty acids have a double bond, or more than one double bond, and this gives them their kinked structure. When you have a kinked structure, you have fewer van der Waals interactions, and therefore you have a more fluid membrane. And so more unsaturated fatty acids equals increased fluidity. Next question. Can ion channels move ions against an electrochemical gradient? And no, they cannot, because ion channels are only involved in facilitated diffusion, which is the movement of molecules down an electrochemical gradient with the help of a protein. And the last question, will a transmembrane protein inserted into the apical surface of an intestinal epithelial cell diffuse in the plane of the plasma membrane to reach the basolateral surface of the cell? And the answer here is no. It's free to move around the apical surface, but tight junctions prevent it from diffusing to the basolateral surface. That's it for the practice questions regarding the plasma membrane.